Hello dear viewers, myself Nasir and welcome to Mr.Zoologist YouTube channel. Today we are going to discuss about Golgi apparatus in detail. So structure of Golgi apparatus and its two models will be the core matter of discussion for the whole lecture of today. Camillo Golgi proposed the structure of Golgi apparatus in 1898. He tells us about the structure of the Golgi apparatus like that it is a membrane bound organelle in most of eukaryotic cells. These are membrane bound organelles which are sac like. Just like might have seen in your life that what is a sac. A sac is a container or a vessel or uh, equipment or cloth like made up of whose one end is closed and one end is open. So here these sacs are open on the both sides. They are found in the cytoplasm of plants and animal cells. Golgi complex is composed of stacks of membrane bound structure. These structures, membrane bounded and protected structures, are known as cisternae. As you can see in diagram here in detail, that these are the membranes on the outer side of these stacks, and each stack is known as cisternae. In a stack, there are 4 to 8 cisternae. Each cisternae is a disc enclosed in a membrane. It possesses special enzyme of Golgi which help to transport of modified proteins to their destination. The flat sacs of cisternae are stacked and is bent and semicircular in shape. That is somewhat of their symmetry of structure is explained about the Golgi apparatus. The cisternae may vary in number from 4 to 8 in animal cells and from 10 to 24 in plant cells. They are equally spaced in a pile so that they are nearly parallel to one another. They are free of ribosome and have swollen ends. The membranes of one end of stack is different in composition and thickness to the other end. One end of the stack is known as the sys face. It is the receiving department which receives the proteins, while the other is the trans face with the shipping department. The sys face is closely associated with the endoplasmic reticulum. As here you can see that the two faces are clearly mentioned. On the downside is the cis Golgi network, that is the receiving face, and on the opposite side is the trans Golgi network TGN, that is the transporting face of the Golgi apparatus. And now if we discuss the structural components of the Golgi apparatus, these are tubules and vesicles. Tubules are short structures arise from the periphery of the cisternae. Some of these enlarge to form vesicle that lies near the end and concave surface of Golgi complex. These are pinches off from the tubules of cisternae. And if we talk about the transfer of material, Golgi apparatus is a major site of synthesis of carbohydrates and sorting and dispatching station. The vesicles that leave the RER and transported to the cis phase of the Golgi apparatus for modification, where they fuse with the Golgi membrane and empty their contents into the lumen. Once inside the lumen, molecules are modified, then sorted for transport to the next destinations. The two models will be discussed in detail will be the cisternal maturation model and vesicular transport model. COP2 coated vesicles move materials from the endoplasmic reticulum forward to the ERGIC and Golgi complex. The ERGIC is the intermediate compartment situated between the endoplasmic reticulum and Golgi complex. COP is an acronym for coated proteins. COP1 coated vesicles move materials in a retrograde direction in the opposite direction from the ERGIC and backward to the Golgi stack in the backward direction towards the endoplasmic reticulum. And second is from the trans Golgi cisternae to backward to the cis Golgi cisternae. So first model is discussed was the vesicular transport model, and now is the cisternal maturation model. The cisternal maturation model envisions a highly dynamic Golgi complex in which major elements of are organelles. Cisternae are continually being formed at the cis phase and dispersed at the trans phase. In this model, the fusion of COP2 vesicles from the ER and ERGIC begins the formation of the first cis cisternae of the Golgi stacks, which later to become TGN. cisternae. Once mature, the TGN dissolves to become secretory vesicles. While these occur, COP1 vesicles continually recycle the Golgi specific proteins by delivery from older to younger cisternae. That is a cisternal maturation model. According to this model, each cisternae matures into the next cisternae along the stack. As you can see in the diagram, first of all is the cis cisternae that transfers into the then into the medial, then into the trans, and then ultimately into the TGN, trans Golgi network. And these fusion occurs within themselves. Each cisternae physically moved from cis to the trans phase and changing in composition as it progresses from one network into the other network. As you can see, that is a flow of the material from the cis phase to the trans phase in the cisternal maturation model. These cisternae mature and transfer from one form into the another form of the cisternae. So next is the vesicular transport model. From the mid 1980s to the mid 1990s, the maturation model Golgi movement was replaced by an alternate model that is known as vesicular transport model, which proposed that cisternae of Golgi stack remains in a place as a stable compartment. They are not moved, they are not maturing into the other, they are at their fixed position. Proteins are delivered from endoplasmic reticulum to the cis phase of using COP2 vesicles, coated protein 2 vesicles, and then progress toward the trans phase of the Golgi apparatus. So in this model, cargo or secretory vesicles that might be the lysosomal enzymes and that may be the membrane proteins, they are shuttled or moved through the Golgi stack from cis Golgi network CGN to the trans Golgi network TGN. In vesicles that bud from one membrane compartment and fuse with the neighboring compartment or further along the stack. As you can see, the materials are not moving from the midway or not by the fusion. The material is moving from the tip to the tip of the cisternae, from one cisternae to the other and up to the transport portion, that is the TGN. So that we have discussed it earlier. Now we discuss the glycosylation in the Golgi apparatus. So glycosylation is the controlled enzymatic modification of an organic molecule, especially a protein, and that modification is done by the addition of a sugar molecule. So there are two types of glycosylation: N-linked glycosylation and O-linked glycosylation. The N-linked glycosylation begins in the endoplasmic reticulum and continues in the Golgi complex, whereas the O-linked glycosylation takes place exclusively in the Golgi complex. N-linked glycosylation is also called co-translational modification. That acronym is CTM, co-translational modification. O-linked glycosylation is also called post-translational modification, therefore it is known as PTM. Carbohydrate processing in the Golgi apparatus complex is called terminal glycosylation to distinguish it from core glycosylation that occurs in the ER, endoplasmic reticulum. It occurs in the cis medial and trans Golgi compartments after secondary proteins traffic from the endoplasmic reticulum. So that glycosylation simply means attaching sugar to the something. That here glycosylation takes place on the protein molecules. So proteins in the lumen of the endoplasmic reticulum and in the ER membrane are transported to the Golgi complex, which is a stack of flattened membranous sacs. Golgi complex has two principal roles. First, carbohydrate units of glycoproteins are altered and elaborated in the Golgi complex. The O-linked sugar units are fashioned there, and the N-linked sugars arriving from the ER as a component of glycoprotein are modified in many different ways. Here you can see different enzymes are attaching the sugar molecules like alpha-mannose enzyme, glucosidase and nucleic acid transferase 1. These are present at the different cisternaes of the Golgi complex, and they are attaching N-acetylglucosamine sugar, mannose sugar, galactose sugar, salicylic acid sugar, and fucose sugar. These sugars are added respectively one by one, and these are carried out by respective enzymes at these cisternae. So glycosylation process continues in the Golgi apparatus. As newly synthesized soluble and membrane glycoproteins pass through the cis and medial cisternae of the Golgi stack, most of the mannose residues are also removed from the core. Oligosaccharides and other sugars are added sequentially by various glycosyl transferases, which are the enzymes present at the respective cisternaes. In the Golgi complex, as in the rough endoplasmic reticulum, the sequence in which sugars are incorporated into oligosaccharides is determined by the spatial arrangement of the specific glycosyl transferases that come into contact with the newly synthesized proteins as it moves through the Golgi stack. So what are the steps in the glycosylation of a typical mammalian N-linked glycosaccharide in the Golgi complex? Following the removal of the three glucose residues, various mannose residues are subsequently removed, while a variety of sugars such as N-acetylglucosamine, galactose, fucose, and sialic acid are added to the glycosaccharide by specific glycosyl transferases. These enzymes are integral membrane proteins whose active sites are face the lumen of the Golgi cisternae. They are facing inside. This is only one of numerous glycosylation pathways. So that was the video. Hope so you have learned a lot about it. We will discuss later lectures in the upcoming videos. Thank you very much.